In the last video, we finished by discussing innate immunity or non-specific immunity, and that includes any kind of defense that can defend you against multiple sorts of pathogens and that you are born with. You don't have to acquire. And so it includes things like your skin and mucous membranes and hair, other physical barriers. Cells that are phagocytes, like neutrophils and eosinophils and macrophages, things that will eat all kinds of different pathogens and defend you. Natural killer cells, which perform immune surveillance and will destroy lots of kinds of abnormal cells. Also interferons, that chemical help signal that your cells send out when they might be, for instance, infected by a virus, and they can slow the spread of disease and increase your resistance to viruses. We also talked about systems like the complement system, which really can be both innate and adaptive, but specifically the alternative pathway of the complement system is innate or nonspecific. Inflammation is nonspecific, right? That dilation of your blood vessels in response to histamine that causes more blood to flow in and therefore more defensive cells and flushing out pathogens, etc. And fever, where you increase the set point of your body's temperature from 98.6 to something higher, like 100 or 101, and that can inhibit pathogens and occasionally accelerate repairs by increasing metabolic processes. But now that we've done a quick review of non-specific innate immunity, let's talk about specific or adaptive immunity. And this is coordinated by those lymphocytes that we talked about earlier, your T cells and your B cells. And this has to be acquired, right? That's why it's adaptive, right? You're not born with this stuff. It has to be acquired, and it's very specific because these cells, each individual T cell or B cell will only respond to the pathogens that have antigens on the outside of their cells that fit with the antigen receptor that these T or B cells have. And there are two main ways of acquiring adaptive immunity. One is called active immunity. This is exposure to the antigen. And you, through exposure to that antigen, that molecule on the outside of a pathogen, like a bacterium or a virus, have an immune response. And you make antibodies, etc., etc. You can also get it passively by receiving antibodies from somebody else. Right? So in active immunity, you have to be exposed to the antigen. And there are really two ways that you can be exposed to the antigen. It can be active naturally acquired. That means you were just exposed to the virus or the bacterium, and you had an immune response, and now you have a memory of that, and you're immune to that from here on forward, at least for a given amount of time. Maybe you got measles, or maybe you got coronavirus, for instance, and you fought it off, and now you have antibodies to it and memory cells, and you can fight it off more effectively in the future. That's naturally acquired because you got infected with the pathogen. But active immunity can also be artificially induced, and this means your exposure to the antigen happened in a medical setting. Essentially, you got a vaccine. And vaccines traditionally contain dead or inactive pathogens or some sort of artificial antigen that they put in your body, and it fools your body into thinking you've gotten that pathogen, right? But you have been exposed to that antigen, to that shape, right? And it causes your body to have an immune response without ever having to be infected, which is obviously pretty nice. Now, there are different kinds of vaccines now that people are developing for the coronavirus specifically that have never been developed before. For instance, shooting a certain type of new genetic material into your cells so that your cells make the antigen themselves and send it out into your body. And in that way, your lymphocytes get exposed to those antigens and cause your body to have a big immune response. And there are a few other interesting ways too that we don't know if they'll work. We'll see. But anyway, a vaccine is a medically induced exposure to an antigen so that you don't ever have to get infected with that pathogen. You still have an immune response. In passive immunity, you don't have an immune response because you were never exposed to the antigen either naturally or artificially. You receive somebody else's antibodies. You did not produce the antibodies yourself. And this can happen naturally, most notably through transfer of maternal antibodies across the placenta. So if you've got a fetus inside you and you're a mother-to-be and you are making your own antibodies, that fetus is getting your antibodies passively. And that's natural, right? Same with breastfeeding, right? One of the advantages of breastfeeding your baby is that they get a lot of your antibodies passively through the breast milk. But you can also artificially give antibodies. And this happens a lot for diseases that act quickly, so your body can't make them very quickly. So like rabies. Rabies is a fast-acting virus, and it essentially crawls toward your central immune system. And once it gets there, you're toast, right? Now we have a vaccine for rabies now. But if you are not vaccinated or your pet's not vaccinated, if you catch it in time, they can give you artificially produced antibodies or antibodies produced in another animal, and those will fight off the rabies virus before your body can begin to mount its own immune response. So that's artificially induced. Or sometimes these pathogens aren't alive, right? Toxins, venoms from snakes, for instance. Those are molecules that are antigens that you can fight off with antibodies. But obviously snake venom moves way too fast for your body to mount an immune response. But you can have antivenom, which really are just antibodies produced by some other animal or artificially that you get shot into your body that help you fight off that venom, those toxic molecules. So within adaptive immunity, active immunity means that you were exposed to an antigen and started making your own antibodies, either naturally because you got the disease or artificially because you got a vaccine. Passive immunity means you're getting antibodies that you did not make, either naturally through like breastfeeding or through a placenta, or artificially because you went to a doctor and they gave you a shot full of antibodies. There are four main properties of adaptive immunity or specific immunity, and those are specificity, number one, meaning those T and B cells have receptors for one specific antigen. And those T and B cells, their responses are activated only toward that particular antigen. So if you have a T cell that's been waiting for you to get H1N1 because it's got an antigen that T cell reacts with, when you get H1N1, you're going to fire up your immune system only to fight H1N1 flu, right? You're not going to fight off a different flu, right, with that same immune response. So it's specific. But it's also versatile because while each individual T or B cell is specific to just a single pathogen, a single antigen that you would find on a pathogen, you have millions of different lymphocytes, and each of them is sensitive to a different antigen. And so, odds are decent that you will be able to mount an immune response once you detect that you're infected with that particular antigen. And then these cells will clone themselves. They will multiply and multiply and mount a large response. In addition to being specific and yet versatile, they also form an immunological memory, and this is really, really essential for adaptive immunity. In addition to fighting off the pathogen right when you get it, right, you also produce memory cells that allow you to, instead of having one or two T or B lymphocytes looking for that kind of antigen, you have lots that then stick around your body, and the next time you get infected with that particular antigen, your body detects it very quickly, and you have a very, very forceful response. And so often you are effectively immune from that disease. And lastly, your adaptive immune system must be tolerant of your own cells, meaning those antigen receptors on the outside of their cells should not bind to antigens that you have on any of your cells, right? They ignore your self cells and only target non-self cells and toxins. when this goes wrong, then that can cause diseases of your immune system. Let's talk a little bit about the process of how this works, how your body responds. So first, you are infected with something, right? Could be a virus or a bacterium, something like that. And you have both B and T cells that are on the prowl, and also macrophages or phagocytes. The antigens that are on the outside of here, right? So the red things here or the blue things here and here, right? Those are the antigens. B and T cells have antigen receptors that are looking out for those specific things. If a B cell finds its appropriate antigen, then it will be primed for activation. Same with the T cell, except that the way a T cell usually comes into contact with its antigen is not on its own. It's with the help of an antigen-presenting cell, or in other words, a macrophage who eats it and then goes and tells the T cell what it ate. It's actually kind of grisly, right? A macrophage will eat up, a bacterium or a virus or something, and will take those bits of the virus, those antigens, and display them on the surface of their cell in a frame called a major histocompatibility complex. They will then display that to the T cell, and if they find the right T cell with the right antigen receptor, that T cell then gets activated, starts making copies of itself. It will then do the final activation of the B cell, which will then make copies of itself, and also the B cells make antibodies. And between direct cytotoxic T cell intervention and also antibodies marking these things for destruction, and T cells, helper T cells, coordinating the response of lots of other types of your immune system, you end up destroying those antigens. And I mentioned that frame with which we hold those antigens is called your major histocompatibility complex. There are really two kinds of major histocompatibility complexes, MHC1 and MHC2. MHC1, all cells have, at least all nucleated cells. Blood cells don't have them, but all of your normal nucleated cells have them. And they essentially are a place where we take just a specific set of the proteins that we produce and we display them on the outside of our cells. And this is important because we display them for immune systems, right? And say, hey, we are a normal, healthy cell with normal, healthy proteins. And if we're not a normal, healthy cell, if we have a viral infection, for instance, in our cell, that means we're making different proteins because we've got viral DNA. And those proteins that we display are going to be a little bit different. And our immune cells, like our natural killer cells or cytotoxic T cells, are going to recognize that and destroy us before we can spread the virus further. Or if we're cancerous, same thing could happen, right? That means we've had a mutation, so we have different DNA, therefore slightly different proteins. Therefore, what we display in our MHC1 is going to be a bit different. So here you can see we've been infected by a virus. That virus causes us to make different proteins. Those proteins get picked up and put in our MHC1, and that protein is abnormal and will get recognized, hopefully, by a cytotoxic T cell or a natural killer cell. MHC2, or major histocompatibility complex 2 proteins, are only found in the membranes of those antigen-presenting cells, right? Those macrophages that eat up pathogens and cut them up into pieces and display those parts on the outside to helper T cells. So here we have a macrophage that's eaten a bacterium, broken it up, taken some of those proteins, and placed them on the outside in that MHC2 to display to a helper T cell to see if it happens to be the antigen that helper T cell is looking for. And this is an actual image of a microscope of a helper T cell and of a macrophage doing this little dance, and this is showing the helper T cell what it's found. And we're going to dig into that process of antigen recognition a little bit more in the next video.